Perfect. If I can, yes, I can advance the slides. All right, so in our discussions about what we might try to do today, hopefully we can kind of keep it informal. Like I said, I have some specimens and I also have some slides. These are some slides that I use with my students who take fungal ecology with me at St. Catherine University. So once we hit past the introduction slides and into the lichen slides, we'll use some terminology that you may be familiar with. We'll talk about the growth forms of lichens, so crustose ones and foliose ones, fruticose ones. We will learn some technical terms that'll help us navigate through a taxonomic key, which if anybody has been working with the lichens of Minnesota or some of the bigger lichen books, you probably will have encountered a key like this. So we'll look at some of these structures, we'll point to them on the lichens so that we know what we're looking at. And then we'll try our hand at a super quick taxonomic key to see how that works. And this happens really fast. It's very free form. It's very interactive. So I want you to know that even though I use this with my students in class I don't lecture either. We stand around with each other, we scratch our heads, we look at specimens. It's very interactive. So that's what we do when I'm at St. Catherine University, which is where I teach, and the microbiologist there. So most of my students are either biology majors and they're taking my class because they really like fungi, or they're nursing students and they're taking classes because they like microbiology. It's going to help them in their nursing degree to know something about microbes. So the nursing students, we talk mostly about bacteria. We talk about diseases. We talk about a lot of blood and guts, and they love it. And with the biologists, we spend more time digging in the dirt, grabbing tree branches, bringing them into the lab. And I also am a molecular biologist. So we are looking at organisms and then we squish them up and we try to learn about how they work by working with their DNA and their proteins. So we have a lot of different ways to interact with the world of biology. Okay, so I really love lichens, and it was a short journey to loving lichens. I have to admit though, growing up in the Twin Cities, I did not know what a lichen was, mostly because I didn't see them very often. And it wasn't until I was working at this camp that we can see a little bit of in kind of central Minnesota when I was in college. Actually, it was when I was a camper at this camp. This is near Nisswa, near Cross Lake. It's called Star Lake Camp, one of my favorite places on earth. And everybody who was with me on this camping trip, the first thing that we noticed is that the tree trunks looked really different than in the Twin Cities because they were covered with all this green stuff and gray stuff. And we said to ourselves, are these trees sick? Because that doesn't make sense because the air out here is cleaner than in the Twin Cities. We had no idea what we were looking at, right? And some staffers told us, oh, these are lichens. And that's where I first met the lichens. So flash forward maybe 10 years and suddenly I'm on staff at this camp. And what I want to do with my time is learn the names of everything. And I started with the plants. So summer number 1, bunch of plants. Summer number 2, we're digging through the kind of bottom of the plants. We're starting to look at mosses and ferns and running out of plants. Summer number 3, I asked myself, well, what can I look at? And the answer is, okay, I'll start to learn the lichens. I think that'll take about 2 weeks because I think there are about 10. And that was wrong. There are a lot. There are a lot. And after that, I ended up at Wolf Ridge, which is where I met Joe. And I came across this cross-country ski boot box herbarium with lots of lichens in it. And I started to look at the lichens. They're pretty good in the cross-country ski boot boxes, but better in person. And I really realized how much I wanted to learn about lichens and how hard it was going to be. And because there's just so many. So that's how I ended up at the University of Minnesota. Here we're looking at the Bell Museum. When I was there, the Bell Museum was much smaller than this. And I was working in the lichen herbarium. I worked with Cliff Wetmore, who was one of the people who really spent a lot of time naming and figuring out where all the lichens in Minnesota were and what their names were. So this is where I really fell in love with, I guess we'll call it lichen taxonomy or academic lichenology. So this is inside the Bell Museum. They have a room that's just full of these cabinets, and the cabinets are full of folders, and the folders are full of sheets. And if you look on the sheets are full of envelopes, and those envelopes are full of lichens. These are collected from all over the world. Cliff Wetmore was a prolific collector. He went to, I think, 6 or maybe 7 continents collecting lichens. And so this is where I got my start as a lichen taxonomist. For my project, I went out to the Smoky Mountains because that's where there were some lichens that we weren't really sure how many there were under the same name. And I collected a whole bunch of lichens. I ran around in the woods. It was fantastic. I brought them back to the lab. I sliced them up and diced them up. I sequenced their DNA, and I came up with an answer to my question. How many different lichens are under the same name? And the answer was there were 3 different lichens in one name. And so that meant that I got to name 2 of them because they were brand new. So these are my favorite lichens. I know they're brown and crunchy. They are, they're charismatic if you look carefully at them under the microscope for about 2 years. But they're in the genus Sticta. They're called Sticta because on the underside, they have this beautiful, fluffy black or brown tomentum and these pits, but they're named after those structures. And you can see them on the bottom here. I don't know if you can see my cursor, but do you see how there's these like little craters that are kind of orange looking? Those are the cyphalae that Sticta are named after. So since I'm the one who discovered these in the first place, I got to name them. I named the one on the left Sticta carolinensis after the Carolinas where I collected them. And then I named the one on the right Sticta fragilinata. So fragilinata means it has the chemical fragilin, which is the thing that makes it orange. So I spent quite a lot of time with real chemists trying to figure out the name of that chemical so I could give the name of the chemical to the lichen. So it was a long process. It was a fun process. I learned a lot, and it brought me out of the woods into the lab, but I was okay with that because it was all in service of figuring out these lichens. So here's my favorite lichen genus still, Sticta. It's a beautiful genus of lichens. It's especially common in the tropics, and I hope that I get to see tropical lichens someday. So these days, I still work with lichens. My students and I, when we get a chance, we're out in the woods, we're collecting lichens, still kind of sort of related to Sticta. Mostly it's Peltidra these days. Joe has been a great collaborator. He is got his nose to the ground in the woods, and he's always looking for patches of beautiful Peltidra lichens that are this kind of unusual green. So most of the Peltidras are big and they're brown or they're cyanobacterial. These ones are special to me because they're green, they have a different photobiont, they do different weird things. And as someone who studies evolutionary biology, like I'm really curious about how they got there. So we spend time in the woods, we find different lichens, I take them back to the lab, I squish them up and I run them through these machines. So we've got some PCR machines so I can sequence their genes. We've got an even fancier over here machine that's even smaller. It's a genome sequencer. So I can know all the sequences of all of the genes in a particular organism, and I can start to ask really cool questions. So that's what I do. I'm most excited about that. Before the pandemic, I was set to go to South America, to Chile, to the driest desert in the entire world. So these are really harsh conditions. Not a lot of things can live there, except for some lichens and except for some algae that also are in lichens, but they can live by themselves. They can choose to live either with the fungus or without the fungus. And so a group and I are waiting to get on the plane when the pandemic is over and someone gives us a little bit of money for the plane tickets, to go back to this desert and to try to understand when the algae have to make a deal with the fungus in order to survive, and when they can get by without the fungus. This is a really, really harsh desert. Like I said, it's the driest desert in the world. It rains once every 18 years to 20 years. And so there's not a lot that can live there, but lichens can live there. The interesting thing is what you can't see at the bottom of this hill is the ocean. So every night, if it's cool enough, a little bit of fog rolls partway up these hills. And then when the sun comes out, the fog evaporates. So it's not like there's no water. It's just that it's only fog. And so different lichens can live kind of at the base where there's a little bit of fog. Different lichens live up at the top where there's hardly any fog or maybe no fog. And understanding like where's the trade-off where the algae can be without the fungus and there's enough liquid for them to just get it from the air, and when do they have to make a deal with the fungus in order to survive because there's not enough water. So the inset here that we're looking at, this is a lichen. Actually, this is not a lichen. This is the algae just by itself. It's really stressed out right now, and so it's making a lot of this orange sunscreen in order to be able to survive without dying. And right over here is a lichen. It has as its photosynthesizer this same algae, but when this algae is protected by the fungus, do you see how it doesn't have that orange pigment anymore? It doesn't need its sunscreen anymore. So it's kind of not invested the energy to make the sunscreen, and you can see its green color underneath it. The green color is still up here, it's just covered by the orange as a protection. And here it's the fungus that's protecting it, and so it can happily photosynthesize. There's a lot of cool questions that we can ask about what happens inside the lichen, because we've got a really stressful gradient from the ocean all the way to the top there. So these are some of the questions that I like to think about when I think about lichens. And I'm going to pause there for a second in case people want to ask things before we start looking at some slides about terminology.
I actually have a question. This is Cheryl. How old are some lichens, like where you're talking about in Chile there? Are they very ancient lichens?
We think so. They grow really, really slowly. So if there's a lichen and it's, you know, maybe 2 inches across, we can guess that that's maybe 200 years old, maybe more, because they're growing so slowly, it's so stressful. And it means whenever the crew is out collecting, we have to be really gentle with absolutely everything that we do, because it's a very delicate soil crust. And so whenever we're walking in the exact same footsteps that we walked in last year, the year before, every other time that we've been there. We don't get to go off that trail because we don't want to accidentally step on something that's been trying to live for 200 years or 500 years and just with a twisted ankle to change its path in life. So they're really old. It's fascinating. I have other questions.
I was just curious. I have a copy of the Lichens of North America, and I don't see your sticta in there. And is that because you discovered it after the book was published?
Exactly. So my sticta was officially published in 2004. And so it didn't make the first edition. And so the next edition that comes out, whenever that is, should have both Sticta fragilinata and Sticta carolinensis in there. But if you like surfing on the internet, you can go to, it's the Consortium of North American Lichen Herbaria. And they have, it's a network of all the different lichen herbaria, like the Bell Museum. And, you know, there's Arizona, there's all the big universities have probably an herbarium, and most of them will have a lichen herbarium. They've started to take pictures of all of their holdings and put them on the internet along with the label data. And there's where you can see some other people's collections and also my collection. So Sticta fragilinata and Sticta carolinensis.
Cool, thanks.
Yeah, it's a fun question. Are there other questions before we head on to some terminology?
Why don't you go ahead, Tami?
This is a trick I've learned as a person teaching in front of a classroom, is I let the silences get really long and uncomfortable. And then there's always one person in the back who will raise her hand at the very end. All right, so maybe that'll be after we see a couple slides. So what I thought we'd do now is to look at some terms that some of them might be familiar and some of them might be new. But if you are, for instance, looking in a book like this and you get to the page of the genus that you like the best, there's going to be a taxonomic key, or like a decision tree, is what I tell my students, that will help you get from sort of a picture or a description to a scientific name. And it's going to ask you a bunch of questions. It's going to ask you about growth form. It's going to ask you maybe about colors or chemistry. It's going to ask you maybe to slice your lichen and look at the different layers on the inside. And then based on your answers to that, it'll lead you to a name. So that's a taxonomic key. And we're gonna just talk about some terminology really briefly. So I think maybe some people, or maybe many people here have heard these terms: crustose, foliose, and fruticose. So I think the thing that we see most often in the Twin Cities are foliose lichens. These are ones that they tend to grow on the bark of trees. We don't have them very often on rocks just because there's enough pollution that the rock growing ones we tend not to see. We call them foliose because they're supposed to look a little bit leaf-like. So the ones that we're looking at here in the center of the screen, they're about as big as my hand, and they're like a piece of paper on a branch, or a piece of paper kind of on the rock. They've got some 3D structure. You can maybe imagine you could put your fingernail under the edge and kind of lift it off a little bit. So the foliose lichens are supposed to look a little bit leaf-like, so folio, like leaf. Fruticose lichens are ones that have more of a 3D structure. I like to think of them either as, if you hold your hands like this, they're either growing up from the soil, or they're maybe hanging down from a tree branch, and they're more of a 3D lichen. So I'm going to hold up a branch, and I hope you can see it in my picture. So most of these things that you see on the branch, these are all considered foliose lichens. They look kind of like tumbleweeds, I think. So those are some pretty common growth forms for fruticose lichens. So fruiting is a word that we use fairly commonly in mycology. We tend to use it to mean anything that's making its a structure that's holding spores. So the fruticose lichens that we're seeing here, I bet people have seen this little cladonia before with its little red apothecia. This is considered a fruticose lichen. We call it fruticose because it's fruiting, or it's making its spore-bearing structures. In this case, these are apothecia on the top of those codicia. So fruticose lichens. And then over here, this is the one that my students have a little bit of trouble with. So crustose lichens, I tell them that they look like paint on the bark of tree or on rocks. They have hardly any 3D structure at all. They still have sort of little dots. Those are the spore-bearing parts, the apothecia. But you couldn't really put your fingernails underneath them and get them off the substrate. So crustose, foliose, and fruticose. For my students who are looking at tree bark for the first time really closely, everything that's foliose looks crustose to them, right? But I bet where you're at, you've got a lot more lichen diversity, and crustose, foliose, and fruticose are things that you're probably pretty familiar with. Okay, so I'm going to pause there. If anybody wants clarification. Because after this, we start to head into the real anatomy and morphology of lichens. Let's see.
Okay, now I'll go.
I have a quick question.
Yeah.
Can a lichen be both crustose and foliose? I'm thinking of a rock posy.
Yeah, for something like that, rhizoplaca, we have a bunch of weasel words that we use. We could say subfoliose to mean that it's foliose, it's on a rock, but it's kind of, the word we would say is closely oppressed to the rock, you know, and it's maybe just peeling off a little bit at the edge. In general, we would say the rhizoplaca would be crustose, but I know exactly what you mean. They're juicy crustose, and they tend to break off a little bit. So these are, I think, helpful first categories. You can also get subfruticose. You can also get lichens that are morphologically plastic, is what you would say. So in some conditions, they're more tightly oppressed or stuck to their substrate, and then in other conditions, they're more relaxed. And that can lead to a little bit of confusion if it's foliose or if it's starting to look a little bit of fruticose. So yes, there's lots of weasel words that you will find in keys to get at that idea that there's some variation, and that foliose and fruticose, it's more of a gradation. But for most of the things that I think you'll see first off, they're more in the core of that. So foliose things will kind of look like this. There'll be a thallus that's round, or maybe kind of more feathery looking at the edge, but it won't be standing up straight like this. Did that answer your question?
Yes, that answered it.
Okay. All right. So a little bit about structures. So when I'm in the lab with my students, we go out, we collect things, we get something like this if we're really lucky. More often in the Twin Cities, we'll end up with something that's more, looks more a little like this. You can't see, there you go. Something that looks like this. And the first thing that happens is the key will ask them a color of a lichen. That's great. They can say it looks gray or it looks yellow. And then it's going to ask them about the risings. And this is where things get a little bit complicated for them, because it means they have to take something like this, they have to lift up and look at the undersurface. I do this all the time just by habit, because the most interesting part of Sticta are the details underneath the thallus surface. So it's going to ask you to look at the risings. The risings are specialized fungal hyphae, so it's the fungal partner of the lichen. We didn't really even talk about what's inside a lichen, did we? The fact that it's a fungus and an algae, or maybe a cyanobacteria, that are living together to make this 3D structure. And most of the lichens that we'll see are stratified. So they're like a fungus sandwich. They've got a fungus cortex at the top, like a skin. Then they have some algae in the middle with fungal hyphae all around them, kind of protecting them, and also giving them nutrients, also taking the sugars that they're making. So the center part of the lichen, or the algal layer, is actually an algal and fungal layer where all the exchange is happening. So carbon in the form of carbohydrates is coming from the algae to the fungus, and then nitrogen and phosphate and all the other things that the algae would need is coming from the fungus into the algae. So that's like the interaction zone. Underneath that is like a little fluffy bed of fungal hyphae. It tends to look white, but sometimes it can have different chemicals on it. That's called the medulla. And then underneath the medulla, we've got another layer of fungus called the lower cortex. And I told you all that so that we could get to here, which is growing out from the lower cortex of the fungus are these risings. So they look like roots. They're kind of named as if they were roots, because maybe the early lichenologists thought that they were roots. They're really actually just helping the lichen hold on to its substrate. So they're helping it hold on to soil or hold on to bark, maybe hold on to moss. And the different shapes of the risings are mostly useful for us as human beings as something that we can look at and use to distinguish lichens from each other. So it's not that more surface area means that it's getting more nutrients from the soil, because that's not where lichens are getting their nutrients from, right? They're getting their nutrients from the alga that's inside. So your key might tell you to look at them and to try to distinguish if you've got something that's really simple like this. It's just a one sort of set of hyphae, it's not branched or anything, coming up from the lower surface. It might ask you, are your risings, are they divided? So they look more like branches. Are they really kind of hairy looking? Or maybe they're more fibrous. They look like someone took a piece of rope and pulled it all apart, right? So there's lots of tiny strands coming together into a big strand. So the technical details, I think, is not something that I ever encourage my students to memorize. This is something they can always find in the glossary of their key. But just to know that when someone's asking you to, or the key is asking you to look at the risings, this is what it's asking you to look at, the bottom part of the thallus, or the body of the lichen, and to closely examine the structure so that you can use it to navigate your key. So for this, if you have a hand lens, I think it's really useful. If you have like a dissecting microscope, that's super useful too, right? You might be asked to look at the top structure, and this, oh, my students are all relieved when we're looking not at the bottom, but we're looking at the top again, because it's easy to see. And it might ask you to look for purina. So does the top look kind of frosty, or maybe, I think frosty is a good explanation. So can you see my cursor? Yeah. So at the edges here, do you see how it kind of looks like a window on a cold day, and it's got that little icing, or maybe it kind of looks like salt evaporating from a cookie sheet or something like that? That's called purina. And again, it's interesting to us mostly because it helps us distinguish one species from another species. Some species have it, some species don't. So it's a diagnostic characteristic. I think this is a great picture of purina. This is, it can sometimes be subtle to tell what's going on here. But I think if you're looking especially at the tips of lobes, it will be easy to see the purina. Back on the bottom side, there are not very many lichens that have these structures called pseudocyphally or cyphally. Yeah.
Do you have a question?
Yes.
Does the purina have a function?
Well, you know, some people suggest that it might be kind of overflow chemicals. So what we do know is that they're chemicals called secondary metabolites. So they're like, if the fungus has extra energy, this is one place that that extra energy can go into making this kind of specialized, we call it secondary metabolites or optional metabolites. So we know that they happen when the fungus has enough extra energy. And beyond that, people have lots of ideas about what it could do. So some people might say that if these chemicals are particularly maybe bitter or toxic, they could be kind of anti-herbivory, but most lichens don't have them. And so that's just somebody's idea. Somebody would like to test that, right? Mostly, if they do have a function, we don't know yet because there are not enough scientists who are able to find a situation where yes, purina and no purina, and the lichens are close enough that you can test them and say, do slugs find them equally tasty? If I expose them to light, do they grow equally well? If I expose them, I take away the light and it's dark, do they grow equally well? So if this is something that fires your imagination, this is an open area of research for you. That's a great question.
Thank
Cyphele?
you. Go ahead.
You're welcome. Yeah. So Cyphele and pseudocyphele are something else that you'll see commonly in keys. So what we're looking at here is the bottom side of a lichen called Pseudocyphalaria. The whole genus is named after the fact that the bottom side has these little polka dots. So what these polka dots are breaks in the lower cortex or the lower skin of the lichen. And what we're seeing is the center part of the lichen, the medulla, the kind of fluffy fungal hyphae, breaking through. Does it have a purpose? Some people say it's so that air can get into the center of the thallus more. Mostly, we don't know what the purpose is, but we do know that the things that tend to have pseudocyphele on the bottom like this are related to each other, and mostly they belong to this genus. There can also be pseudocyphele on the top surface as well. If there's a cortex or a skin that continues from the lower cortex down into the pit, we would call that cyphele. So real cyphele have a cortex. Pseudocyphele, they're just kind of holes that the medulla or the fluffy layer is breaking through. So this is what it looks like when we're looking at the actual thallus or the body of the lichen. If you were to take a cross section and look at it under the microscope, it might look like this. So here you can see what we're looking at. These are fungal hyphae, these little bits right here. Here's fungal cortex. So it's still fungal, but it's growing in a different way. The fungal hyphae are kind of glommed onto each other. They're kind of glued together to make that lower skin. And then there's a break in the lower skin, and you can see these fluffy medulla hyphae breaking through. Why do we care about this if we're out in the woods? It's because it helps us identify lichens down to genus or maybe down to species.
I have a question. Another.
Yeah.
Is it a misprint, or do you really mean morphology as opposed to morphology on that?
I really mean morphology. It should be morphology, yes.
Thank you.
Morphology is not a thing as far as I know.
I was worried it was a special lichen term.
There's so many special lichen terms. It's hard to know, isn't it? All right, here's morphology. It's got the R back. Whew. And we're looking at cilia now. So cilia along the edge here, it's the same word as the structures that some of my students see when they're looking at pond microorganisms. There's some structures called cilia that help those organisms swim. These are a different kind of cilia. My students who are taking anatomy and physiology they say, cilia, we know what that are, your eyelashes are cilia. These are like eyelashes, right? And the answer is, in the sense that they're long and skinny, yes, they're cilia. So it's a word that we reuse as biologists to mean kind of long, skinny thing. So they're not used for swimming, they're not used for keeping stuff out of your eyes, but they are long and skinny. And here we can see them along the edge of the thallus. So here we're looking at the upper cortex of a lichen. We're looking at the edges, and do you see the little projections here? These are the cilia. So they're separate from the rhizines that are the sort of holdfasts. They're along the edges here, and they're really prominent, right? Over here, we're looking at diagrams of them. You can see that they can sometimes be long and really prominent. Sometimes they're shorter and they're around the edges. So why do we care? Again, just because it helps us work through our key. Okay, I've got one more slide, and then we have a chance to maybe look at some lichens and put some of these terms to use. Are we ready for one more slide? I see Joe's nodding. Joe's ready for the slide. Everyone else is like, oh, there won't be a test, will there? There will not be a test. All right. The last thing we need to know are about the asexual and the sexual structures. I like this slide because all the information is on one page. It is a little complicated though. So we're going to split it and we're going to look at the left-hand side first, and then we're going to look at the right-hand side. The left-hand side is showing what happens with asexual reproduction. So the fungus that gets dispersed or kind of sent away from the parent thallus here, it's genetically identical. Same thing with the algae. The algae that are in those little kind of Ceridia, they're genetically identical. So this is a type of we can call it fission maybe, or asexual reproduction. It's just little chunks of the lichen that get broken off, and they're kind of shaped so they're really good at dispersing away and finding a new home and maybe starting a new lichen thallus or a new lichen individual. So what we're looking at here, we're looking at a cross section through a foliose lichen. We can see all the layers. We can see the top or the upper cortex. We see the algal layer that we were talking about. We can see the fluffy hyphae of the medulla starting to wrap around each of these algal cells. And then we see this lower cortex or the lower protective layer of fungus. And we've got some rhizines here that's going to help this thallus hold on to the substrate. And then we see that there's a break in the upper cortex, and some little packets of algae wrapped around with little fungal hyphae are escaping out of the middle there. So if it's a windy day, they might blow away. If it's a rainy day, they might get splashed away and carried to a new place. So each little packet of algae and fungus together, it's called a ceridium. And because it's Latin or Greek, and we have to be complicated about how we make singular and plural, if there's more than one, we call them ceridia. So we see a whole bunch of ceridia escaping right here. And in a key, you'll be asked to look at the shape of the place that's broken. And that place in the cortex that's broken is called a ceralium. It's going to ask you, are the ceridia in a ceralium around the edges of the thallus? Or maybe it's all over the thallus, or maybe it's just in the center parts, or maybe it's at the edge of a tip. So the placement of the ceralia, which are kind of the patches of ceridia, is also something that's diagnostic on your key. I'm going to stop there and ask if there's questions before we head over to the apothecium, which is our last set of terminology.
About the fungi algae combination.
Say it again.
And whether or not they exist independently or they only exist in collaboration with each other in a lichen. And so are the fungi a species, are the algae a species, or is just the lichen a species?
These are the questions, that people were really asking themselves in the 1950s and 1960s. And it caused a lot of knockdown, drag out fights in the lichen community, because people who were in the desert, for instance, were looking around and they would see a lichen and they'd see right next door an alga, and they'd say, this is the same algae. I see this orange trentipolia on this cactus spine, and right on that same cactus spine, I see a lichen. Inside, it's got trentipolia. And using microscopy, they looked similar enough where people said, yeah, okay, maybe they're the same thing. The complicated thing was, as soon as the algae is inside the lichen thallus, the fungus is doing some things that causes the algae to behave differently. So the algal cells get bigger. They sometimes start to break apart. So they don't have the same kind of connection between each other. If they reproduce sexually inside the lichen, they tend not to. And so there was always the possibility that we're actually looking at different species of the same algae. So maybe it could be that one species of algae in this genus can be free living and a different species has to be lichenized. So people thought about this for a long time. And the same thing with the fungus. And so just looking through the microscope, the jury was out. And then came the DNA revolution, where you could just take a chunk of the lichen thallus, squish it up, get a sequence of DNA and match it. You can say, I have a chunk of DNA from this lichen and the algae inside. I have a chunk of DNA from this algae outside, and they're identical. So I know it's the exact same algae inside. And what people found is there were some genera of lichens where the exact same fungus looked totally different. So one of my favorite ones is Sticta and Dendris cacolin. Sticta has a big, beautiful, flat thallus. Dendris cacolin looks really big and like reindeer antler. It's the same fungus. And what's different about them is the algae. And something about the combination of the fungus and the algae makes the fungus do something different. So to answer your question, that was a really long windup, wasn't it? Most of the time, the fungus is only found in the lichen. It's hardly ever, there are very few species of lichenizing fungi that can live without algae. I know of like 3. For the algae, what we think is that they can be lichenized or sometimes they can be free living. That's true for the Trentipolia, like the ones that I was looking at in the desert or hope to look at in the desert. And it's less true for the ones on this stick here. For these, most of them have an algae called Trabuxia, and we hardly ever find them free living. Now that's a big fight too, because there's at least one guy in Austria who says, I have been sampling air, and in my air filter samples, I sometimes find Trabuxia. And we don't know if that's just a ceridium that got in his filter, or if it's really the algae that's living freely. So if you like knockdown, drag out fights, you can still have them. But all of which to say, in general, one lichen is one fungus and one algae living together, with exceptions, a couple exceptions. That's an interesting question.
I have a sort of silly question maybe, but you mentioned that prior to sequencing, so using DNA for sequencing lichen, that they had totally different shapes. And so before the sequencing, were there, I'm sure there were some lichen that were the same, but they were identified as 2 different or named as 2 different species, or have there been any like mergers saying, after the DNA sequencing, and have there been made some corrections like in the ID books or anything like that?
Yes, so it's happened basically every way that you can imagine it. So back in Linnaeus's time, for Linnaeus, he saw basically anything foliose and he just named it lichen. The genus lichen, it's full of everything that I would call a lichen. And once people got a little more sophisticated, they said, actually, if it's a different color, if it's a different shape, it's a different organism. So they had to split that genus into several.
Right.
And there's been some mergers too. So the one where there was the thing that looked like Sticta and the thing that looked like Dendris cacolin, with being the same fungus, that's the thing that caused a big knockdown, drag out fight in the field of nomenclature. Because when people had been naming the lichen, they were naming the association. And after that, it got complicated because people were saying, does the name of the lichen belong to the association? Does it belong to the fungus? Does it belong to the algae? It can't belong to the algae because we already have names for half of these algae. So then the name must belong to the fungus. Except for that, there's 2 different genera named after the same fungus. So what do we do? And it got complicated, but the upshot is that now there's a code of nomenclature called the Botanical Code that covers fungi too. And we've decided that the name of the fungus is the name of the lichen, which means that the genera that have different lichen structures, but the same fungus, it got complicated. So some of those genera had to disappear. So we don't say Dendris cacolin anymore. we say it's the blue-green photomorph of Sticta, or it's the green photomorph of Sticta. So we got more precise, but it got longer.
Thank you.
Yeah, this is fun. I'm having a great time. This sounds like a great time to open up to all kinds of questions. I'm not sure what time we're supposed to end, but I'm here until we're done with questions.
Are there any more questions or?
All right, if there are not any more questions, maybe we can do a couple quick guess and check. So I'll be quiet while you tell me foliose, crustose, fruticose. Do you remember? This was a too easy question, wasn't it?
Oh, how are we responding?
Can we can put it in the chat?
Oh, okay.
Just jump in.
Oops. I have too many buttons. I'm looking for where I can see the chat. Joe, I'm going to call on you. Can you see the chat?
Yes, I can see the chat.
Is there anything in the chat?
Yeah, there's all kinds of good stuff in the chat.
You're going to
And
be
guess
my
what?
you're going to be my
have-
chat monitor.
You have brilliant students. That's what I am deducing from this.
Excellent. So, Joe, I'm going to have you answer the question then.
Oh, well, I'm going to read this. It says A is crustose, B is fruticose, and C is foliose.
All right. Here's a trickier one. We haven't talked about some of these. We didn't talk about Isidia, did we? But we did talk about Apothecia and Ceridia. So Ceridia, you remember, those are the kind of dusty or maybe flower-like conglomerations of a little bit of algae with fungi wrapped around them. Apothecia, we didn't talk about. Yeah, we did. Did we? The sexual structures right next door? We didn't. Let me go back.
Didn't.
We got too excited talking about Ceridia. All right, so Apothecia over here, if we take a cross-section through a lichen, this is the sexual structure of a lichen. So it's the fungus, it's making spores. So each of these spores, it's genetically different from the base fungus here. So if you look sideways through here, you can see each structure here has a packet of 8 spores, and we see these spores being released into the environment. When we're looking head on, we see that apothecium, it looks like a disc. So we have seen some apothecia, right here. There's an apothecium. Over here is an apothecium. They look kind of like little circles. These all tend to have a rim on them. So looking here, here's an apothecium, here's an apothecium. There's all kinds of apothecia on this lichen. Over here, here's an apothecium, here's an apothecium, here's an apothecium. Lots of black apothecia on this crustose lichen. Over here on this fruticose, the apothecia are really obvious. They're bright red. And they're at the top of these structures called pedicia. They're like little matchsticks whose job it is to hold up the apothecia. So if the spores get shot, they can catch the wind and go elsewhere. So now I'm kind of just giving you this answer for free, which is great, right? Because we're at the end of our time. Apothecia, Ceridia, one of these has Isidia too. All right, so Joe, I'm going to ask you again. The freebie for apothecia, which
Yeah.
one has apothecia?
So we all agreed that it's C.
Yep.
The last one over there.
How about Ceridia?
So far, your students aren't writing answers to that, including myself, because I always get them confused. I'm pausing to wait to see what your other students say.
It's tricky, isn't it? So we agree it's either A or B. We're going to pick the one that looks most like little patches of flour. By flour, I mean like the kind you put in your pancakes.
Can't tell. I can't tell at all. If I could flick it. A looks like maybe it flicks off better. Am I?
You're actually on. So. It's A. Those little white flakies you're talking about.
Yeah, we're looking at these structures right here. So
flowers.
What we're looking at here is a ceralium with tons and tons and tons of ceridia on it. So if we were looking with our hand lens, we would see it looking kind of like a pile of flour, or look kind of grainy. And each little grain is those little algal cells wrapped around with a couple fungal hyphae. And they disperse together so the fungus and algae can start a brand new phallus together. So asexual reproduction. And then over here we have Isidia. Isidia, it's the same idea. It's fungus and algae together. The only difference is that instead of just kind of medulla hyphae, or kind of the fungus and the algae together, this also has an extra layer of protection around it. So it's got fungal cortex protecting it. So I like to think of this as like a really skinny part of the phallus that can just break off. So here we've got really tall Isidia around the edge that can just break off.
So Tami, are the Isidia typically on the margin, or it varies maybe?
It varies. You can find them on the center of the phallus too, or kind of spread across the whole phallus. But I would say they tend to be on the edges more, and I don't know why. I will also say that when the phallus cracks, and so like the cortex breaks, you can see Isidia along the break line too.
Okay.
That's my experience. All right.
Okay. I have a question.
Yeah.
I think I heard you say that the ascospores were different genetically than the phallus fungus. How does that happen?
Yeah, so that's sexual reproduction. So it's the same fungus, but it's like the daughter or the son of that fungus. So
where's the partner?
Exactly, right? That's a great question. And what we know from molecular biologists who have been asking themselves this for a long time and have evidence from tools like I use for PCR is that in a lichen thallus, there can be more than one individual fungus of the same species. So we don't use male and female, but you can imagine that there's, the A mating partner and the alpha mating partner, or the A and the B mating partner. They find each other in the phallus. It's the same species, but different individuals. And then they can merge and make that apothecium. And if you remember your biology and you're comfortable with the terms meiosis happens, and that's the independent assortment of chromosomes from the mom fungus and chromosomes from the dad fungus mixing around so that you've got a new combination in each of those spores. That's a great question. It makes it a lot more complicated inside that lichen thallus, doesn't it?
Thank you.
All right, what else would you like to ask? This is where I wish we were really in person, right? So that we could get out our sticks and we could grab some things and play with some things and look at some things.
This question doesn't have to do with morphology. It has to do with going back to the collections that the state has.
Mhm.
How do you feel about what the knowledge base is of the lichens in Minnesota? Has there been the kind of investigation and reporting that really you feel is adequate for the state?
That's a great question. I will say that of herbaria that I've visited all across the world, including Iceland, the National Herbarium there, Panama, the National Herbarium there, other herbaria in the United States like Wisconsin, Duke University, where I was a PhD student. Minnesota is one of the best in the United States. It's one of the best in the world. It has a lot of lichen collections. That's partly because of the originator of the herbarium. It's also because of Cliff Wetmore, who was a fantastic collector. So if you collect 1,000 species in a year and you do that over the course of your lifetime, you might have 30,000 or 40,000 specimens lodged in the herbarium. So Cliff Wetmore, I think he had close to 100,000. So he's like at least two people or maybe three people's worth of collectors. And he's a classical taxonomist. He was great at finding new species and naming new species. So if you're an herbarium collector, the statistics you like to show off are, first of all, how many different specimens you have, how many different places in the world you have really like good collections, like lots of different lichens from, and how many types or how many lichens that were described for the first time, and you've got the actual lichen that was actually named for the first time. And on all those metrics, the University of Minnesota herbarium is like in the top 10 in the world. So it's very good. There's always more to learn though, especially if you're looking at really small things, and especially if you're looking kind of in the northern part of the state, there's a lot of new things to be discovered still.
Tami, I have another question.
Yeah.
Do you use iNaturalist in any? Go ahead.
I do as a consumer, and I'm consuming Joe Olewski's iNaturalist site, which is fantastic for lichens.
Okay.
Hey, everybody. I just want to thank everybody for letting me tag along, and I've learned so much already, and I look forward to continually join. I'm 15 minutes late to my previous engagement. I gotta run. Thank you so much, Tami, and everybody else.
It's great to see you, Joe.
Bye.
What were you gonna ask? I was gonna talk to Joe a minute. Oh.
So if we don't have anything more right now, Tami, when do you think you would like to join us again?
Why don't you tell me when is a good time for you and what you would like to do? I think it'd be great if we can meet in person and actually grab some lichens, slice and dice them, look at them under the microscope, do some chemical tests and things like that. So some of that we can still do over Zoom. It's a little complicated to get a microscope image in real time onto here. It's more doable to do a chemical test. So you can tell me when works for you.
So when are you going to be up this way?
My plan is to have a couple students with me in August, kind of the end of August. I think before the school year starts. That's my plan. I don't know if
So-
that's too soon.
So maybe we could tag on to either end of your trip or during your trip somehow to make an in-person get together.
That sounds great. I would be happy to come to you.
Oh, great. Super. August sometime.
Wonderful.
Yeah, so thanks a lot for joining us, Tami, and everyone else for joining in. And for those of you who are local, we're going to possibly meet out, do an outing next week. I know Steve Schoen's Lichen of the Week is something that we can't bring inside unless we want to carry a tombstone with us. So next week is going to be an outing. So thanks again, everyone.
Thank you so much. It was wonderful to meet you all.
Tammy.
I look forward to
Nice
seeing
to
you in
meet
person.
Bye.
Bye.
Thank you, everyone, for joining us.
Thanks, Cheryl.
That was done. That was done.
Ron Brodkin.
So this is stick to the next.